Let me just quickly make sure no one else has emailed saying they're having difficulties. Okay. All right then. Well, welcome everybody. Excuse me. My name is Sean Krosnick, and I'm at Tennessee Tech University in the Hollister Herbarium. And this is our first coffee with a curator for this academic year. So it's great to see everybody. And we are we are recording this, excuse me, so that we can post it on our website. And I just invite everybody to check out the Society of Herbarium Curators Southeastern Chapters website. We have a lot of good stuff on there, and we're continuing to develop that. So please check it out and stay in contact with us, and hopefully come to the rest of the seminars that we have set up for this semester. And if you have ideas for seminars, please send us those as well, because we'd love to include folks that you guys know about. So today I wanted to introduce our speaker, Rabern Simmons, and he is a mycologist, and I don't think I can pronounce chytridology. Is that correct? Do you want to tell me?
A chytridiomycologist.
Yeah, okay, there you go. That's too many syllables, even for me. So, um, but yeah, so he is the curator of fungi at the Purdue University Herbaria, and he's been there almost 3 years now. And prior to that, he had a postdoctoral fellowship at University of Michigan, and then two other postdoctoral experience, one at University of Maine and then the University of Florida. So he's had a lot of great experience, and I think he's got some really interesting stuff to talk to us about today. So I guess without further ado, I'll let you take over.
All right. Thank you for having me. I am Rabern Simmons. I'm the curator of fungi here at the Purdue University Herbaria. Um, my first thought when I was approached by your chapter to speak was, what are my southeastern United States credentials? But I'm here at Purdue in West Lafayette, Indiana, but I am originally from Big Stone Gap, Virginia, 15 minutes Kentucky, half an hour to Tennessee. So I hope that today at least you will accept me as one of your own. Um, can everyone see my screen, my presentation? Okay, great. Um, so today I wanted to talk a little bit about the Purdue University Herbaria, but then I'm going to completely hijack this discussion and talk about some other very interesting fungi and collections of very interesting fungi as well. So I hope you're on board for that. Um, this is a little bit different than when I give this kind of presentation, though, because I'm actually speaking to people that know what herbaria are, and that's generally not the case. I have to go about defining it and talking about the importance of herbaria. But you probably noticed from the first slide, you may have a question, why am I saying herbaria, Purdue University herbaria, and not herbarium? And there's a very long story behind that. The Purdue University Herbarium has always kind of existed conceptually since the beginning of Purdue University. Classes started in 1874 after establishment in 1869. And within the first 5 years, John Hussey started to take collections of specifically Indiana specimens and collected about 2,000 within those first 5 years. Um, Charles Barnes took over after Hussey. He was actually a teacher at Lafayette High School at the time, but a lot of people are probably more familiar with Barnes's later work at the University of Wisconsin, where he coined the term photosynthesis. And the person that took over after Barnes left was Stanley Coulter. Stanley Coulter was our first dean of the School of Sciences here at Purdue University. He has a building named after him, Coulter Hall, on campus, and actually that's where the herbarium was held for many years in the basement. And the herbarium, upon Coulter's retirement, was dedicated to him. So instead of having just this nebulous Purdue University herbarium, it was actually established as the Coulter Herbarium. but today we call it the Kriebel Herbarium, and that's in honor of Ralph Kriebel. Kriebel was a student of a fashion to Charles Dean. Dean was the first state forester of Indiana. He literally wrote the book on the flora of Indiana, and he had a particular love for ferns and oaks. So much so that there were some very interesting oaks that he found. I think two or three that were going to be destroyed in order to widen a road. And he purchased the land that they were held on for $75 back in 1915 and donated this 0.2 acre plot of land to Indiana to preserve these trees, which he thought at the time were indicative of a new species of oak. It turns out now we know that they are, in fact, hybrids, but still very interesting trees. And this state preserve has been formally recognized as the smallest state preserve in Indiana, the Dean Oak Monument Forest. So, according to the state of Indiana, 3 trees constitute a forest. Um, Dean would inspire Kriebel, who came to Purdue in 1943 as an extension specialist. Unfortunately, he passed away in 1946. But at that time, he had accumulated 10,866 mounted specimens. And upon his death, his mother had those and she wanted rid of them. So she donated them to, or actually, I believe, sold them to Purdue University. And at the time of this donation, it was thought that this was the second largest collection of Indiana plants, second only to Charles Dean. In 1961, the Coulter Herbarium was rededicated to Kriebel formally. And just this past year, I had the opportunity to meet Kriebel's grandchildren. And their father, Robert Kriebel, the son of Ralph Kriebel, was actually a biographer that wrote for the Purdue University Press for many years. And one of his books that he wrote was Plain Old Charlie Dean, about the life of Charlie Dean, who'd inspired his father. So again, why am I talking about herbaria and not herbarium? We also concurrently had the Arthur collection on Purdue campus. That was started by Joseph C. Arthur in 1882. Arthur started working here at Purdue in 1887, and he was a very prominent mycologist and botanist. Specifically, he worked a lot on rust fungi, these obligate parasites of living plant material that have multiple interesting life stages, very confusingly, so much so that he did over 4,000 inoculation experiments to determine how these different fungi were related to their hosts, and eventually would go on to write the Manual of the Rust of the United States and Canada. To my knowledge, he was the only person that has been recognized as the president of the Botanical Society of America twice. And he loved Purdue. In fact, he wrote the song in 1901 called Viva Purdue. You can go to the Purdue University Herbaria website and hear a sample of it on piano. He retired in 1915 and continued to work on his collection. He lived in Lafayette, across the Wabash River from West Lafayette. But after 3 years, he got fairly tired of that arrangement to come to visit his collection that he started before he came to Purdue, and that Purdue really didn't fund. So on the night of July 4th, 1918, in the middle of the night, he had some loading vans brought to campus. He loaded everything up, and he took his ball and he went home. Purdue was not exceptionally pleased with this arrangement, and they contacted the Tippecanoe Sheriff's Office. And eventually, in August of 1918, Arthur and Purdue came to an agreement. The 3 major points of this agreement were: one, Arthur could come and go as he pleased and use whatever equipment he wanted, because Purdue didn't want to lose that credential, their relationship with this very prominent mycologist and botanist. Two, Purdue would pay him $1,000 in 1918 money to essentially buy the collection back from him. This worked out to about 3.5 cents per specimen at the time. And with this money, he took his wife on a tour of Europe. The third point being that he didn't want his collection to be lost, his name to be forgotten, and just subsumed by, at that point, the larger of the two collections, the Purdue University Herbaria or the Coulter Herbarium. Um, so he wanted his collection to be kept as a separate entity, the Arthur Herbarium. And that is why, legally, based on a contract from August of 1918, we are required to be the Purdue University Herbaria and not Herbarium. Arthur would continue to train people in rust fungi in particular. The second director of the Arthur collection was George Cummins. Cummins had a very tumultuous relationship coming to Purdue. He was actually hired to come here from Michigan. There was a mycologist here at Purdue, E.B. Maines, who had been trained by the professor at Michigan, Calvin Kaufman. And Maines asked Kaufman for an illustrator to come and help Arthur with his book. Kaufman sent Cummins to Purdue, and very shortly thereafter, Kaufman passed away. In order to replace Kaufman, the University of Michigan hired Maines from Purdue. So Maines went back to Michigan, kind of just leaving and abandoning Cummins here with, at that point, a researcher that was kind of just wrapping things up, working on his book, but still like he didn't sign on for this. Um, Arthur was never his formal teacher, he or advisor. He had to finagle some things with faculty members, but eventually he did get his PhD here at Purdue, and he continued on working for the university and eventually would become, at this point, the longest serving director of the Arthur collection. He retired in 1971, but continued working. His last published work was when he was 99 years old, and he lived to the age of 102. There is an ongoing belief in the Mycological Society of America that if you want to live a long and prosperous life, you should work on rust fungi, based primarily on Cummins. But also, Arthur lived to be 92. The Cummins was also widely recognized within the Mycological Society of America. He was the first recipient, the inaugural recipient of the Distinguished Mycologist Award, which is the highest honor that is bestowed upon members of that fungi in particular. Eventually, these two collections, the PUR, which is the Arthur, and the PUL, which is the Kriebel, would be merged at least in the same space. The PUR was shuffled into our current building here in Lilly Hall when botany and plant pathology was moved here. There was some reshuffling at one point of all of the non-rust specimens of the Arthur into the Kriebel collection. I'm not sure what sort of arguments there may have been about that at the time, but now we have the Arthur collection as exclusively a collection of rust fungi on obligate plant hosts. That's all that's in the Arthur collection. The Kriebel is everything else, including all the other fungi. The PUL was eventually transferred from the Department of Biological Sciences to Botany and Plant Pathology as well. And in 2009, we moved to our current location, which is a more interior, climate-controlled space in Lilly Hall on the campus of Purdue University. Joe Hinnon oversaw a lot of this reshuffling. He was the director after Cummins. He actually was a student of Cummins and obtained his master's and PhD from Cummins. He retired in 1995 and actually just passed away this past year again at the age of 96, living a fairly long and prosperous life as a rust fungal specialist. Between 1995 and 2012, there was no formal director of either of these collections or the herbaria as a whole at that point. It wasn't until Kathy Ames started in 2012 that changed, and she has just led this complete renaissance of the Purdue University Herbaria, specifically in relationship to the Arthur. Kathy is a mycologist, but definitely one with leanings toward rust fungi. And since she started, the Arthur has grown from 90,000 specimens to 125,000 specimens. And those are just the accessioned ones, not the things that we just have sitting in boxes waiting to be worked on. That number actually may be higher if it wasn't for the fact that for the past 2 years, Kathy has been away in Washington, D.C., serving as an NSF program officer. This past year, she was also the Mycological Society of America president, and I believe she was recently elected president-elect of the International Mycological Congress. So she definitely has a busy schedule, but this place could not be what it is currently without her. The PUL at the moment contains about 110,000 specimens. So that's everything that's not a rust fungus. I can't remember the exact numbers. I think we have, I can't remember the exact numbers on what we have for fungi in particular. But the Arthur collection, like I said, is now 125,000 specimens. And that's including over 3,000 type specimens of rust fungi because Arthur and Cummins and Hinnon did so much work on taxonomy. You can also see from these numbers, Arthur didn't have anything to worry about, at least with Kathy at the helm, about his name being forgotten and his collection being absorbed into the larger of the two collections. His is definitely the larger of the two collections. Our oldest specimen was collected during the first circumnavigation of the globe by Captain James Cook. It was collected in Terra del Fuego, Argentina in 1769. It's a barberry leaf with a little tiny rust fungus on it. It sat in a cabinet for 200 years in England before it was properly examined and that rust fungus was detected. It was sent to Joe Hennon here at Purdue to be verified. And you can see the micrographs on the side that he produced of the spores to confirm, yes, this is a rust fungus. And they allowed him to keep the specimen. So I'd like to say to tours of Purdue University, Purdue University was established in 1869, so this specimen is actually 100 years older than the concept of Purdue University. We also have specimens from various other scientific researchers as well. In particular, I'll point out George Washington Carver. We have about 70 plants and rust fungi that were collected from Iowa and Alabama during the course of his studies. And the work is ongoing. We've had a lot of people in this lab in particular working on fungal taxonomy specifically. Dr. Aim has had a relationship with researchers in Guyana for over 25 years. She has both had researchers go from here to Guyana and brought researchers from Guyana to Purdue to obtain degrees. And we also have researchers that are looking at other Central American taxa and just taxa here in Indiana even. I'll also mention here that Paula is one of our recent graduates with a PhD. She was looking at these larvae that are obligate parasites on the obligate parasitic rust fungi on plants. She was doing molecular analyses of these larvae based on specimens that she collected that had been preserved at the time that the plants had been preserved. So I think this is just an excellent example of the importance of a physical specimen and keeping a physical specimen, because it shows that, you know, if a photo had been taken 50 years ago, this project could not have been conducted. If we needed the physical specimen in order to actually do this process, find these organisms that otherwise would have gone unnoticed. And it really just emphasizes to me the true importance of herbaria. The another project that we constantly have going is the universal rust barcoding. This is just molecularly characterizing as many rusts as we can from this collection so that there is an online database of rust, because a lot of these materials aren't necessarily represented in online genetic libraries. And finally, one project that I've been helming for the past couple of years has been the African Plant Project, where in conjunction with the University of Kansas, where we are forming a large database of the all of the plants taken from Africa that are held in North American herbaria, and forming a database and perhaps returning those at some point. There is also, as part of that, we were recently awarded an REU, so we have a continuing undergraduate research support for that project. Also, as the curator of fungi, I am responsible for the living cultured collection of fungi within the AIM lab group. and we have about 7,000 active cultures. We have them in the Alfred, the AIM Lab fungal record database. And thanks to the work of all these undergraduates and others that aren't pictured here, we have been conducting this 5-year plan to transfer all 7,000 of those in a rotating cycle every 5 years so that we can keep the materials somewhat more fresh than they have been. I know 5 years seems like a long time for various types of fungi, but a lot of these cultures that we're looking at, some of them haven't been transferred in 15 years or longer. But we're slowly trying to get these better managed and properly recorded, but it could not be done without a huge amount of undergraduate assistance. Finally, maybe one of the most important things that we do as herbaria curators is outreach. I try, since I've come here, to do as much as possible and reaching out like I did for this presentation to as many people as I can to talk about the Purdue University Herbaria. Um, in the top left corner, you can see our spring fling activity. Spring fling is a function put on by the College of Agriculture here at Purdue, where we open up labs or areas to the public so that they can come in, see how science is done, perhaps participate in projects. And in this instance, we had children coming in and learning how to preserve specimens and to make mounts so that they can form their own herbaria. On the right, at the top, is function that I went to in Remington at the library in Remington, Indiana, where they wanted to learn about mushrooms, and I just went and talked about various types of mushrooms that we could find in the state and various other locations, and just fungi in general. I've spoken to various visiting academic societies. I'll even go out into the middle of the forest and set up a poster and just talk to whoever comes by. This is at the horticulture park here on campus. It's actually part of a larger event called Fall into Nature. And that was a lot of fun just to get out and just show people what we do. I've also gone to Baylor University and other institutions to talk about both the Purdue University herbaria, but also chytrid fungi or zoosporic yew fungi that is my kind of a research background, and I'll get into that soon. And we were very, very briefly mentioned in the April issue of National Geographic this past year. But that is the first time in the 130 years of National Geographic that fungi have ever been featured on the cover of the magazine, which is ridiculous that it took that long to do. But we are grateful that they finally did it, and they did a very several articles about fungi in that issue. Um, Finally, in the middle here is a letter to the editor that I wrote to our school paper, the Exponents. Um, it was actually started as a letter to the author of an article, and the article was about the importance of cursive handwriting and how a lot of students can't read cursive, can't write cursive. And I was saying, like, thank you for bringing this to people's attention because it's a skill that we really need, especially in the case of trying to digitize our various specimen sheets. There's a lot of information that's probably going to be in some really weird cursive. Um, and I sent this in. They asked if they could publish it as a letter to the editor. It's actually in the issue of the Exponent that goes out to new students last year, and so hopefully some parents saw it as well, and maybe inspired them to get their younger students trained in cursive handwriting. But specifically, the people that kind of unintended that saw it was the Purdue alumnus, the alumni magazine of Purdue University. Purdue University just celebrated 150 years of Purdue. And they contacted me and said, we just did 150, all these stories about 150 people, 150 places, 150 things, but we didn't talk at all about the herbaria, uh, Cummins, Hennon, Arthur, any of that, because we just didn't know about it. So, kind of as an apology piece, they did an entire article about us in their spring issue of the Purdue Alumnus. And I love promoting this one because they make us look so good. it's beautifully written. It's beautifully photographed. Of course, it has a very strong tilt towards mycology and fungi because I was involved with it. Um, but I definitely encourage you. You can find this very easily again on our Purdue University Herbaria website. But I would encourage everybody to check that out.